Bismillahirrahmanirrahim. I welcome you all in this lecture. Today we are going to discuss the last part of dark reaction. The dark reaction is also called dark cycle. It was not invented in or happening in a dark atmosphere, but it is since it's light independent reaction. That's why it is called as dark reaction. Now there are three core components in dark reaction. Number one is carbon fixation, followed by reduction and regeneration. So in order to understand the picture, we need to make a background assessment or a story. So in continuation to the last lecture where we discussed about the light reaction, over here we see that there is a sunlight which is falling on a thylakoid, and the thylakoid is generating two products for us, that is ATP and NADPH. And now we are proceeding to this left side part, and this portion is actually happening in the stromal mass, in the stroma. So inside a chloroplast, the stromal region is the place where Kelvin cycle or a dark reaction cycle is happening. Since it is called the cycle, that means the number of things are repeating again and again, irrespective of whether the light source is present, but it is not dependent on the light circle. Okay, it is happening in the daylight, but it is not dependent on sunlight. So it begins with the step 1. Initially there is a CO2 present, and that CO2 in combination with ribulose biphosphate. Now this molecule is 5 carbon, and the CO2 merge together to form another compound which is called phosphoric phosphoglyceric acid. Now this addition of this carbon with the 5 carbon compound leads to the formation of PGA, and this PGA is further been converted into glyceraldehyde 3-phosphate, or you can say under the influence of ATP and NADPH, which were being generated by the light reaction to turn into a stable sugar molecule. Now this PGA is converted into G3P. A portion of just this G3P, or in short form we call it as PGAL, PGA, PGAL, both are the same thing. So a portion of it is used for the generation of carbohydrate, while rest of the substance goes again in the cycle. That is the simplest understanding about the Kelvin cycle. Now let's differentiate the process step by one. Now over here, the reason of adding the same picture is if we focus more precisely on the number of carbon atoms, then we can understand why these numbers are 3 or different. Now this begins with the carbon dioxide, which is represented over here with 1 dot, followed by ribulose biphosphate, and this ribose indicates that these ribose are 5 in number. So in carbon fixation, this 5 carbon compound, so let me draw this ribulose biphosphate numerical numbers. Let me use another color which is should be, let's say, blue color. So let's assume that this carbon is being used. Now this one is the carbon. Now this ribose, as the name indicates, contain 5 carbons. Now 5 plus 1 is equal to 6. So this 1, 2, 3, 4, 5 and 6. Now this 6 carbon formed compound is unstable. So it is an unstable compound. Now since it's an unstable compound, so it usually gets disintegrated, and this disintegration leads to the formation of two phosphoglyceric acid components. Now 6 carbon branch into 3 carbons, and since these are the 6 carbon atoms, so it has been branched into 3. That's why the number 3 falls over here, but it is basically 2x. So you can write 2x because we are expecting 2 phosphoglyceric acid, phosphoglyceric acid or PGAL compounds at this step. Now once these two three PGAs are formed, then they are under the influence of ATP. Now this ATP is coming from the light reaction we have seen, and this one also, and this ATP and NADPH is reduced. Since I have told you that reduction means that this is the removal of hydrogen or loss of electrons, so NADP is reduced, ATP is gone into ADP, so it's an energy dependent process, and it is formed into a more stable product which is called glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate are formed as 6 in number. In this example, there are 6 molecules. One molecule goes straight away for glucose synthesis. Remaining five compounds containing 3 carbon atoms go all the way to form ribose biphosphate binding protein. Now this biphosphate compound contains how many carbons? 1, 2, 3, 4 and 5. Okay, so all this cyclic movement of carbon is of our interest. So over here we were expecting that the 5 carbon merge with the 1 carbon dioxide carbon, and we form a 6 unstable compound which bifurcates into 3 carbon atoms, which is called PGA, under the influence of light reaction ATP and NADPH. This forms a stable compound which is G3P, and one G3P is gone over here for glucose, while remaining all the five components are going back to that cycle of ribose biphosphate. Okay, and this is again going into the cycle for CO2 utilization, and this process has been differentiated into carbon fixation, reduction and regeneration. So let's draw it in a very simplistic way over here. So the step 1, as mentioned earlier, CO2 carbon along with the 5 carbons mentioned over here in pink color form these 2 compounds, phosphoglycerate or phosphoglyceric acid, both are the same thing. Now this phosphoglycerate is formed, they are 2 in number because this molecule is unstable. Now this molecule in step 2, under the influence of ATP and NADPH, turn into glyceraldehyde 3-phosphate. Now this G3P is converted into glucose. One portion went for glucose, that is C6H12O C6. That is just the general formula for glucose. We already know that C6H12 and O6, that is CH2O divided by the number of N's we have. So it's pretty much the same thing I am trying to write over here. So one thing is done, and this is that this G3P, glyceraldehyde 3-phosphate, converted into glucose, but not all of it. A majority of the portion, which is let's say 90% of the portion, revert back into the cycle for the generation of this compound. So this one, okay, ribose biphosphate compound. Okay, and it's an energy dependent process because it requires ATP to make glyceraldehyde 3-phosphate, and similarly this glyceraldehyde 3-phosphate also require ATP to convert into a ribose formate compound. Okay, that's the simplistic view for what you can say Kelvin cycle. Now in order to understand this image, we can now learn this image more beautifully. This one, this is says CO2 acceptor. 5C means ribose biphosphate compound. Ribose biphosphate compound or 5C atom combined with one carbon turn into 6C, which is temporarily stable, and it reverted back into 3C compound, which is I have written it as PGs, and then this is unstable molecule, but since they are three in number carbon, so we expect 2x. So this step is called carbon fixation because we are adding one carbon during the process of photosynthesis. In next step, under the influence of ATP and NADPH from the light reaction source, the compound is further been stabilized into glyceraldehyde 3-phosphate. Now out of this 3C, 1C goes for glucose production, remaining a majority of the lot goes follow this channel and goes for CO2 acceptor to reinitiate the cycle again and again. So this is called dark cycle or dark reaction cycle, but it doesn't happen in dark. It is basically happening in daylight, but you can say it is light independent reaction. Okay, thank you very much for your time and attention.